Our third experiment is conducted by Alfred Hershey and Martha Chase. That was 1952. They conducted the experiment by using a bacteriophage, and we all know a bacteriophage is a virus that can infect a bacteria or that infects a bacteria. So the bacteriophage is a virus that infects a bacteria. And as you all know from grade 11 that a virus is made or composed of protein capsid and a DNA. So this bacteriophage, which is a virus that can infect a bacteria cell, is composed basically of a capsid, protein capsid, and genetic material, which is DNA. So it looks like that, a protein capsid and a DNA. We also know from chapter 1 in by 12 that protein has a main component of sulfur and DNA has a main component of phosphorus. So I can trace protein by using a radioactive isotope of sulfur, which is the sulfur 35. So sulfur 35 is a radioactive isotope. And sulfur is a main component existing only in proteins. Same thing here, DNA, which is mainly made or has a phosphate group in it. So I can use a radioactive phosphate, so a radioactive phosphate, which is phosphate 32, to trace DNA. So Hershey and Chase, Alfred and Martha, they conducted this experiment into two parts, part A and part B. Here's part A. So they brought the virus, the T2 virus, which is the bacteriophage that infects a bacteria, and they made sure that it is now, I can now trace the DNA in it using phosphate 32. So the DNA in our virus is now traceable by using a radioactive phosphate, which is phosphate 32. They brought that T2 virus that's marked with the phosphate 32, marking the DNA there. So what is it that I'm tracing now? I'm tracing the DNA. And they allowed it in a beaker, in a conical flask, to infect bacteria. After a while, they used a blender to create agitation to separate the virus capsid from the bacterial cells. Why? Because if you do remember that viruses, when they infected the bacteria, that was from Bio 11, okay, chapter 2, that the viruses, they land on a receptor site. So here is a receptor site. So this is our T2 bacteriophage virus, where we labeled our DNA here with the phosphate 32 radioactive. And this virus lands on a receptor site on our bacteria, and it injects its genetic material in it, and the genetic material will basically take control of the cell, and it will make the cell to produce a specific protein that will cause the cell to die at the end. So this viral fragment of the DNA will take control of the bacteria, producing a specific protein that will cause the death of the bacteria cells. So that's why here we use agitation after allowing infection. So I'm going to start here again. So I brought the T2 virus where I labeled its DNA. So the phosphorus here, I labeled the DNA, and that is what I'm tracing now, to infect bacteria in a conical flask. Then I used a blender to separate the virus capsid from the bacterial cell, which were attached on top of it. And then I'm going to do centrifugation. Centrifugation is a high-speed centrifugal force, and it's basically to separate the different materials by their densities, where dense, like basically heavy things, will be settling at the bottom of my tube, and lighter things or less heavy things will be on top. And of course, it goes without saying that bacteria is way heavier than virus. So bacteria will always settle at the bottom with centrifugation, that's separating things by their density. So when I look at here, I have bacteria at the bottom and virus. So I have the bacteria and the virus. Where is the radioactivity tracing coming from? So where's the radioactive tracing coming from? It's coming from the bacteria section. So the bacteria section is actually emitting radioactivity, which means that in this bacteria I have here, so here I have my viral DNA, which I traced in the beginning here by using the phosphate 32. That was part A, and also this part says it very loud and clear that DNA is the genetic material, because DNA, which I traced using phosphate, entered into the bacteria, caused its death.